This protocol is demonstrating that we can now determine the effect of migrastatic inhibitors in detail in 3D cancer models. The main advantage is the ease and all-in-one approach with which this technique can be carried out.
This technique allows the assessment of microstatic drugs in cancer research in a 3D setting, allowing the testing of drugs in a more tumor environment relevant setting.
This method may be applied to testing of cytotoxic drugs in cancer or, for example, to assess the effect of radiation on cancer proliferation and cell migration.
It is useful to practice the medium removal step and collagen replacement with ordinary 96-well plates to become confident. Also, training on a confocal microscope is essential.
Visual demonstration of this technique is critical for researchers to understand the required delicate handling of the spheroids and their use in confocal microscopy.
On the first day of experimentation, have the standard culture medium, in this case U251, ready for the appropriate cell line. In a tissue culture hood, add 0.5 milliliters of trypsin into the culture to trypsinize the cancer cells. Count the cancer cells on a hemocytometer and dilute to a concentration of 5 × 10⁵ cells per milliliter. Keep the cell suspensions in clearly labeled sterile universal tubes. Resuspend the cells by gentle inversion to avoid cell clumping. Use a pipette to add 200 microliters of the cell culture to each well in a 96-well plate. Add 200 microliters of 1x PBS to each empty well to avoid evaporation. Incubate the cells in an incubator at 37 degrees Celsius. After 24 hours, check the cells by bright field microscopy.